We found a sample, and it was, again, we're exploring in places where there were rivers and where there were lakes, and much as on Earth.
That might have a biological origin, but requires more data or further study before reaching a conclusion about the presence or absence of life.
In 1976, a NASA experiment on Mars returned results that, on Earth, would have confirmed the presence of living organisms, and then the agency buried it for decades. That statement sounds like conspiracy. It isn't. It's the conclusion of the scientist who designed the experiment, a man who spent the rest of his career trying to get NASA to listen. In 1976, NASA's Viking landers touched down on Mars, carrying biology experiments built to answer one question: is there life on the red planet? One of those experiments, the labeled release test, returned a clear positive, not ambiguous, not marginal, positive. The kind of result that, if it had come from any soil on Earth, would have been published, peer-reviewed, and accepted without a second thought. But NASA didn't accept it. Instead, the agency spent the next 4 decades designing missions that carefully avoided running the same test again. And the scientist behind that experiment, Gilbert Levin, died in 2021, still insisting his instrument had done exactly what it was built to do: detect life. Now, nearly 50 years later, a different rover on a different part of Mars has found something that makes Levin's claim almost impossible to ignore. And what it found is forcing the entire scientific community to confront a question they've been sidestepping since the summer of 1976. To understand why this story matters, you need to go back to the summer of 1975. At the Kennedy Space Center in Florida, 2 identical spacecraft were being prepared for the longest journey any American mission had ever attempted. Viking 1 and Viking 2 were, at that time, the most ambitious planetary missions in history. Each consisted of an orbiter and a lander, and each lander carried a suite of instruments specifically designed to search for microbial life on Mars. The biology package included 3 separate experiments, each designed by a different team. The gas exchange experiment, led by Vance Oyama at NASA's Ames Research Center, would look for gases released by organisms incubated in Martian soil. The pyrolytic release experiment, designed by Norman Horowitz at Caltech, would test whether Martian organisms could absorb carbon from the atmosphere the way plants and cyanobacteria do on Earth. And then there was the labeled release experiment, the brainchild of Gilbert Levin. Levin was an engineer and microbiologist from Baltimore who had founded a company called Biospherics Incorporated. His background wasn't in planetary science. It was in detecting contamination in water supplies. That practical, applied science mindset shaped the experiment he built for Viking. The concept was elegantly simple. You take a sample of Martian soil, you add a drop of nutrient solution, water laced with simple organic compounds, each tagged with radioactive carbon-14. Then you wait. If anything in the soil metabolizes those nutrients, it will release gas containing the radioactive carbon. A Geiger-style detector will pick it up immediately. On Earth, this method was extraordinarily sensitive. It could detect as few as 30 living cells in a soil sample, far fewer than any other technique available at the time. His co-experimenter was Patricia Ann Strott, a molecular biologist who had been an assistant professor at Johns Hopkins University before making the leap into space science. Together, they refined the labeled release instrument through years of testing, calibrating it against soils from deserts, volcanic fields, and Antarctic dry valleys, the most Mars-like environments they could find on Earth. The launches went smoothly. Viking 1 landed on July 20th, 1976, on the flat, rock-strewn plains of Chryse Planitia. Viking 2 followed on September 3rd, touching down at Utopia Planitia, roughly 4,000 miles to the north. The lander's cameras sent back the first photographs ever taken from the surface of another planet. Rust-colored rocks scattered across an ochre plain, a sky the color of butterscotch, tinted by fine dust suspended in the thin atmosphere, a landscape that looked like the driest, coldest desert imaginable, but emptier and infinitely more still. Now imagine the scene inside the Jet Propulsion Laboratory in Pasadena, California. It's late evening. The fluorescent lights buzz overhead. Banks of monitors flicker with green-on-black data streams. Paper printouts spool from dot matrix printers curling onto the linoleum floor. The air is stale. Recycled ventilation mixed with the smell of cold coffee and nervous sweat. Dozens of scientists and engineers are crowded into the control room, some standing, some hunched over terminals, all of them staring at numbers that are about to either confirm or deny one of humanity's oldest questions. Nobody is sleeping. Nobody wants to miss this. On Sol 10, the 10th Martian day after Viking 1's landing, the labeled release experiment ran its first cycle. That evening, around 7:30 p.m., Patricia Strott was at the computer terminal. Levin stood behind her, along with several other team members. She worked the keyboard, entered the command, and hit the print button. The computer printed out the data points from the first 9 hours of readings. The curve climbed steadily. Strott looked at it, looked at the team, and said 4 words. "Oh my God, it's positive." The initial results were exactly what a biological response would look like. But what came next was far more complicated than anyone expected. The data was unambiguous. The Martian soil had consumed the radioactive nutrients and released radioactive gas. The curve on the printout climbed in a smooth arc, virtually identical to what you'd see testing fertile soil from a backyard garden on Earth. But the Viking team had built in a critical safeguard. A second soil sample was heated to 160 degrees Celsius, hot enough to kill any known terrestrial microorganism, and then given the same nutrient solution. That sterilized sample produced almost no reaction. The curve stayed flat. This was the textbook signature of biology. Active soil responds, dead soil doesn't. But the team went further. They heated a third sample to just 50 degrees Celsius, warm enough to stress living organisms, but far too cool to destroy most chemical compounds. That moderately heated sample showed a significantly reduced response. Not zero, but weakened. On Earth, that pattern has one explanation. You've damaged a population of microbes without fully killing them. They ran the experiment again at the same site. Same result. They ran it at Viking 2's landing site, 4,000 miles away. Same result. In total, Viking performed 9 labeled release cycles across both landers. Every active sample responded. Every heat-sterilized control did not. The results were consistent, repeatable, and exactly what the experiment had been designed to detect. If this data had come from soil in Antarctica, or from deep ocean sediment, or from a cave in the Atacama Desert, the paper would have been written within weeks. Life detected. Case closed. But this wasn't Earth. This was Mars. And within days, a different instrument on the same lander delivered a result that threw everything into question. The complication came from the gas chromatograph mass spectrometer, the GCMS. This instrument had one job: detect organic molecules in the Martian soil. Carbon-based compounds, the molecular building blocks that every known form of life on Earth is constructed from. Proteins, lipids, amino acids, DNA, all organic. If there was life in the soil metabolizing nutrients, there had to be organic molecules present. That's not speculation, that's biochemistry. The GCMS found none, not a trace. At the parts per billion level, the Martian soil appeared to be utterly, impossibly sterile. Cleaner than any soil on Earth. Cleaner, in fact, than the lunar samples brought back by Apollo astronauts, and the moon has no life at all. This created a paradox that tore the Viking biology team apart. Levin's experiment said something in the soil was alive and eating. The GCMS said there was nothing organic in the soil to be alive. Both instruments were working correctly. Both datasets were genuine. They couldn't both be right, or so it seemed. The biology team leader, Harold Klein of NASA Ames, ultimately sided with the GCMS. His official conclusion, which became NASA's official position, the labeled release results were caused not by biology, but by some unknown chemical oxidant in the Martian soil. Perhaps peroxides or superoxides, created when ultraviolet radiation from the sun, unfiltered by Mars's thin, ozone-free atmosphere, struck the iron-rich regolith and produced highly reactive compounds that mimicked a metabolic response. Levin pushed back hard. He pointed to the 50-degree heating test. A robust chemical oxidant wouldn't be weakened by a temperature that barely warms a cup of tea. Biological organisms would. He pointed out that no one had identified the hypothetical oxidant. No one had reproduced the Viking results in a lab using any combination of non-biological chemicals. The data fit biology. The data did not fit chemistry. And the GCMS, he argued, may simply not have been sensitive enough. Its detection threshold could have allowed as many as 1 million bacterial cells per gram to go completely unnoticed. Nobody could prove him wrong, but almost nobody in a position of institutional authority was willing to agree with him either. There's no easy way to say this, but what followed was not a scientific resolution. It was an institutional decision dressed up as one. NASA closed the book, and for the next 3 decades, the agency moved on. The silence from NASA didn't mean the question went away. It went underground. In 2012, an international team of scientists, including Levin, published a peer-reviewed analysis using a mathematical technique called cluster analysis, a method for detecting patterns in complex datasets. They reprocessed the original Viking-labeled release data using modern computational methods unavailable in 1976. Their conclusion, published in the International Journal of Aeronautical and Space Sciences, the Viking results were consistent with biological activity and inconsistent with the non-biological alternatives proposed over the preceding decades. The paper didn't make headlines. It should have. Meanwhile, on Mars itself, a new generation of robots was quietly building a case. In August 2012, NASA's Curiosity rover landed in Gale Crater, a 96-mile-wide impact basin straddling the Martian equator. Curiosity carried the most sophisticated mobile laboratory ever sent to another planet. It wasn't designed to detect life directly. NASA had learned that lesson, or at least had decided the question was too fraught to ask head-on. Instead, Curiosity was built to assess whether Mars had ever been habitable. But one of its instruments found something no one had predicted. The tunable laser spectrometer, part of the sample analysis at Mars Instrument Suite, detected methane in the Martian atmosphere. The amounts were tiny, background levels averaging about 0.4 parts per billion. But over 5 Earth years of continuous monitoring, spanning 3 full Martian years, the team documented something remarkable. The methane concentrations rose and fell in a clear, repeating seasonal cycle. Higher in late northern summer, lower in winter. The same pattern every year. The results were published in the journal Science in June 2018, led by Chris Webster of the Jet Propulsion Laboratory. On Earth, over 90% of atmospheric methane is produced by biological activity, microbes in wetlands, organisms in ocean sediments, bacteria in the guts of animals. Geological processes can produce methane as well, through a reaction called serpentinization, where water interacts with certain iron-rich rocks. But the seasonal variation Curiosity documented was roughly 3 times larger than any known geological mechanism could account for. Webster stated it plainly in the paper. The seasonal pattern was real, and a biological source could not be excluded. Then there were the spikes. Sudden, unexplained methane surges where concentrations leaped to about 7 parts per billion, nearly 20 times the background level. These bursts appeared and vanished unpredictably. One planetary scientist at NASA Goddard, Mike Muma, voiced what many researchers were thinking privately. He said the explanation in the back of everyone's mind is biological activity. You'd expect life to be seasonal. If you're finding this fascinating, a like truly helps this channel continue investigating these mysteries. The Curiosity team didn't hold a celebration when the methane paper came out. There was no fanfare, just a growing, uncomfortable realization that the question NASA thought it had answered in 1976 wasn't answered at all. At this point, the researchers assumed they had their answer, or at least, they had permission to stop looking for one. They were wrong. Because in July 2024, a different rover, on a different part of Mars, found a rock that changed the entire conversation. NASA's Perseverance rover had been exploring Jezero Crater for more than 3 years. This 45-kilometer-wide basin had been confirmed by ground-penetrating radar to have once held a deep lake, fed by rivers that carved channels through the crater rim more than 3 billion years ago. The rover had already collected over 24 rock and soil samples. Some contained interesting mineral signatures. None were extraordinary. Then Perseverance drove into Neretva Vallis, the ancient river valley on Jezero's western edge, a channel roughly 400 meters wide, once a powerful waterway carrying sediment into the lake. There, in the dried riverbed, partially buried in mudstone, the rover encountered an arrowhead-shaped rock about 1 meter long. The team named it Chayava Falls, after a waterfall in Arizona's Grand Canyon. The rock's surface told a story you could almost read with your eyes. White mineral veins, evidence that water had once flowed through cracks in the stone. Small dark flecks, 100 to 200 microns across, scattered like pepper across the surface. The team called them poppy seeds, but the features that made everyone stop were larger. Pale centered spots ringed by dark borders, arranged in concentric patterns across the mudstone. They looked like the rosette markings on a leopard. Someone said it, the name stuck. Perseverance's Sherlock instrument, a Raman spectrometer that uses an ultraviolet laser to identify organic molecules, scanned the rock and detected carbon-based compounds in the mud. It was the mission's strongest organic detection to date. Science team member Melissa Rice later said the finding made them pause. Was this rock worth one of their limited, irreplaceable titanium sample tubes? The answer was unanimous. This was exactly the kind of organic-bearing, fine-grained sedimentary rock they had traveled to Jezero Crater to find. They drilled a core, sealed it, named the sample Sapphire Canyon. Then they aimed Pixel at the leopard spots, a precision X-ray instrument that can map chemical composition at the sub-millimeter scale. What Pixel found in those spots would take a full year of peer review before anyone was ready to say it out loud. This is where the story pivots from what was found to what it could mean. And the chemical fingerprint inside those leopard spots has forced scientists into a conversation many of them would rather not be having. Pixel's analysis, led by planetary scientist Joel Hurowitz of Stony Brook University, was published in Nature on September 10, 2025. After 12 months of independent peer review, the paper revealed two specific minerals inside the leopard spots. The dark rims contained vivianite, a hydrated iron phosphate. The pale interiors contained greigite, an iron sulfide. Both minerals are well documented on Earth. Both are commonly associated with one specific phenomenon, microbial metabolism. On Earth, vivianite typically forms in waterlogged sediments, peat bogs, and around decaying organic matter. Greigite is produced by magnetotactic bacteria, organisms that harvest energy from iron-sulfur chemistry. When you find these two minerals together, arranged in concentric reaction fronts alongside concentrated organic carbon, the most common explanation on this planet is biological. Microbes consumed the organic material, used iron and phosphorus and sulfur as metabolic fuel, and left behind these mineral patterns like chemical fingerprints pressed into ancient mud. The Perseverance team spent a year trying to construct a non-biological explanation. They modeled high-temperature alteration, the idea that volcanic heat could have cooked the minerals into this pattern. It didn't match. The surrounding rock showed no evidence of extreme heat. They considered acidic fluid interactions. Perhaps corrosive water flowing through the mudstone produced the spots chemically. But the mineral assemblage didn't fit an acidic environment. They tested abiotic redox chemistry scenarios, reactions between the sediment and oxygen-poor groundwater. Closer, but still incomplete. No single abiotic model could account for the combination of vivianite and greigite arranged in reaction fronts alongside concentrated organic carbon. Katie Stack Morgan, the mission's project scientist at JPL, put it directly at the September 2025 press conference. Abiotic explanations are less likely given the paper's findings, though they cannot yet be fully ruled out. Hurowitz was equally measured. He told the Planetary Society that the team knew right away there was something genuinely unusual in these rocks. But knowing something is unusual and being ready to call it biological are two very different things in planetary science. And this is the detail that keeps researchers up at night. The Shayava Falls rock wasn't just the first place on Mars to show signs of organic molecules. It wasn't just the first place to show minerals associated with microbial metabolism. It was the first single location to contain all three ingredients that life requires simultaneously. Evidence of liquid water, a chemical energy source, and organic carbon, all in one rock, all preserved for more than 3 billion years. Some researchers still urge extreme caution, and they have reason to. The ALH 84001 meteorite in 1996 appeared to show nanoscale Martian fossils, and even President Clinton held a press conference about it. That claim was later undermined by alternative explanations nobody had considered at the time. The scientific community learned a painful lesson about premature announcements. One interpretation, controversial but difficult to dismiss, is that the same institutional caution born from ALH 84001 has now delayed the recognition of genuine evidence by decades. The pendulum swung so far toward skepticism that it may have overshot. But consider what 5 decades of Mars exploration have produced, taken together. Viking's labeled release experiment returned consistent positive results for metabolism at 2 sites 4,000 miles apart. Nobody has replicated those results using non-biological chemistry. Curiosity found seasonal methane whose variation exceeds any known geological explanation, behaving the way biological emissions behave on Earth. Perseverance found organic molecules embedded in ancient lake sediments alongside mineral assemblages that, on this planet, are produced almost exclusively by microbial activity. The researchers tried every non-biological explanation they could devise. None of them adequately worked. Each of these discoveries was made by different instruments on different missions at different locations on Mars, yet they all point in the same direction. If we take this evidence at face value, the implication is extraordinary. Mars was not merely habitable in its ancient past, it was inhabited. If that conclusion holds, and confirming it will require returning the Sapphire Canyon sample to Earth for laboratory analysis no rover can perform, the consequences extend far beyond planetary science. If life arose independently on 2 planets orbiting the same star, from the same raw materials, in the same solar system, it would transform our understanding of biology's place in the cosmos. It would mean that life isn't something that happened once, by accident, on one lucky planet. It would mean that wherever the conditions converge, liquid water, chemical energy, organic molecules, life emerges, not as an exception, as a default setting of the universe. And given that astronomers have identified thousands of exoplanets in habitable zones across our galaxy, with hundreds of billions more estimated to exist, the implication is staggering in its simplicity. We would not be rare. We would not be special. We would be one example of something that happens everywhere. Of course, there are simpler explanations that remain untested. There may be geochemical processes unique to Mars that no Earth laboratory has replicated. The honest scientific position is, we don't know yet. But the fact that NASA used the phrase potential biosignature in an official announcement, a first in the agency's 67-year history, tells you how decisively the evidence has shifted the conversation. Gilbert Levin died on July 26, 2021, at the age of 97. He spent the final quarter of his life writing papers, giving talks, and appearing on podcasts and radio programs, all making the same argument he had been making since the Carter administration. His experiment detected life on Mars, and nobody with institutional power had been willing to say so. He wasn't bitter exactly. He was relentless. On a program called The Space Show, in one of his last public interviews, he said something that stayed with everyone who heard it. He said he was certain that NASA knew there was life on Mars. He called for a re-examination of the Viking data by an independent panel, scientists with no career stake in the agency's official position. That panel was never convened. Patricia Strott walked a different path to the same conclusion. For years after Viking, she refused to endorse Levin's interpretation. She was cautious by training and temperament, a molecular biologist who understood better than most that extraordinary claims require more than one experiment's worth of evidence. But as the decades passed and more data streamed back from Mars, subsurface water, preserved organic molecules, seasonal methane that no one could explain, she reconsidered. In a conversation published by Scientific American, she said something remarkable for a scientist of her rigor. She had come to agree with Levin. The Viking experiment found life on Mars. Neither of them lived to see the Shyava Falls discovery. Neither read the Nature paper. Neither heard a NASA official describe leopard spots on a Martian rock as potentially the strongest evidence of ancient life ever found beyond Earth. There is something worth sitting with in that. Two scientists spent their careers arguing that their data was being ignored. They published papers, they gave lectures, they appealed to panels and review boards. They watched mission after mission launch to Mars: Pathfinder, Spirit, Opportunity, Phoenix, Curiosity, Perseverance. And not one of them carried a life detection experiment. Not one was designed to answer the question their data had raised. The scientific community chose to look for water rather than look for life, because looking for water was safer. Nobody's career was destroyed by finding water. And then, decades after both scientists had been sidelined by the consensus, a rover found a rock that matched exactly the kind of evidence they always said would vindicate their work. The irony is almost unbearable, but the sample they spent their lives arguing for, the kind of evidence they always said was needed, sits right now in a sealed titanium tube on the surface of Mars. Sample number 25, Sapphire Canyon. If the Mars sample return mission moves forward, and as of now, it remains in the planning phase, caught between budget pressures and technical challenges, that tube could reach an Earth-based laboratory within the next decade. Once here, scientists could examine its mineral structures under electron microscopes, sequence any preserved organic molecules, and perform isotopic analyses that no rover instrument can match. The kind of work that could settle the question once and for all. Consider what that rock has survived. It formed more than 3 billion years ago, when Mars still had rivers and rain and a thicker atmosphere. It solidified at the bottom of a lake that no longer exists, on a planet that was slowly losing everything: its atmosphere, its magnetic field, its warmth, its water. It endured the slow death of a world. It lay buried under sediment for eons, then was exposed by wind and erosion. It withstood billions of years of cosmic radiation, bombarding a surface with no ozone shield. It outlasted every dynasty, every empire, every civilization that has ever risen and fallen on Earth. And now it sits in a tube the size of a marker pen, drilled by a 6-wheeled robot, in a crater where the nighttime temperature drops below -100 degrees Celsius, and the air is less than 1% as thick as ours. Somewhere in that tube, in a few grams of reddish-brown mudstone, there may be proof that we were never alone. Not fossils visible to the eye, not bones or shells or anything that dramatic. Just a quiet arrangement of iron and sulfur and phosphorus and carbon, Minerals left behind by something that, over 3 billion years ago, was eating and breathing and living in the mud at the bottom of a Martian lake. Something microscopic, something ancient, something that, if it existed, would redefine what it means to be alive in this universe. The data doesn't care about our comfort. Multiple independent lines of evidence, gathered by different instruments, on different missions, across different decades, on different parts of Mars, are converging toward the same conclusion. That doesn't mean the question is settled. It means the question has changed. It's no longer whether life could have existed on Mars. It's how much longer we can avoid confirming what the data already suggests. So here's the question that won't go away. If a NASA experiment returned positive results for life on Mars in 1976, and the scientific establishment spent nearly 50 years constructing alternative explanations rather than running the test again, what does it mean that the evidence has now returned, stronger, from a completely different location, using completely different instruments, pointing in exactly the same direction? Is this a story about a discovery being made? Or is it a story about a discovery being made twice, because the first time, nobody was willing to accept it? Or is it possible that the hardest part of discovering life beyond Earth was never the science at all, but the willingness to believe what the science was telling us? If this changed how you see what's happening on Mars, share it with someone who needs to hear it. The samples are sitting on the surface right now, sealed in titanium, waiting for a ride home. Gilbert Levin waited his entire life for this moment. He didn't live to see it.
The only question left is whether we'll finish what he started.